So we'll take a look now at the more complicated figure of the life cycle of a non-vascular plant. And I just want to remind you that these figures in your text are going to get really, really complicated. And so it's good to just always touch base with the bones. And so that's what I'm going to emphasize is kind of the bones, the bare bones of this figure. So remember that we have spores, and I'm going to start with the spores again. So that's where I started before. The spores will go through mitosis. They're already haploid, so I'll add that term. Haploid spores will go through mitosis to develop into gametophytes. And so for this type of a non-vascular plant, there's actually a female gametophyte and a male gametophyte. Those will grow and grow and grow into multicellular gametophytes. In certain regions of the gametophyte, specialized tissue called either archegonia in the female gametophyte or antheridia in the male gametophyte, some of the cells will mature into gametes. So in the antheridia, some cells will mature into sperm. In the archegonia, some cells will mature into an egg within each one. And then if there's water available in the environment, the sperm that are out in the environment will be able to swim over to the egg inside of the female gametophyte. That produces the diploid zygote. Notice that the diploid zygote is embedded into the female gametophyte tissue. Okay, this allows the gametophyte tissue to feed the zygote and the growing embryo because the diploid tissue is non-photosynthetic. So now the zygote will start to go through mitosis. So here it is in the embryonic form. Again, it's inside of the archegonium and it's receiving food from the female gametophyte. And this will continue to grow and grow and grow into the sporophyte. So the diploid sporophyte grows out of the gametophyte tissue. Within the gametophyte tissue, usually at the tips, you will find sporangia. So sporangia are specialized structures that produce spores. So within the sporangia, the cells will go through meiosis, and each cell that goes through meiosis will produce many, many spores. The sporangia will oftentimes break open somehow and release those spores into the environment. We think of those spores as offspring because those spores are what can now go out into the environment, land on soil, and make a new gametophyte. So the advantages of the sporics life cycle, especially when we compare it to the algae, resources from one generation, from the parent generation, helps the next generation. So this is that matotrophy that I was talking about. The zygote develops into the embryo, all while being retained in the maternal tissue. So the maternal tissue, the parent, is feeding nutrients to the offspring. And so that's really, again, allowing resources to be shared. All the life of the mother is able to help give a boost to her offspring. The other benefit of the sporic life cycle is that there's a much greater amount of spores, so a much larger spore production. So again, with algae, remember, we can only get 4 spores per reproductive event, but for the sporic life cycle in plants, we get hundreds of spores per event. And so that allows much larger dispersal of offspring into the environment. That advantage is so strong to having a sporophyte that produces lots of spores that actually over plant evolution, we see the sporophyte become larger and larger. So the more sporophyte you have, the more spores you can make. That's like the kind of brief summary there. So the bigger the sporophyte, the more the spores. And so in the non-vascular plants, we have a relatively small sporophyte and it's short-lived. So the gametophyte is photosynthetic and it produces this very small, spindly, short-lived, like it's usually only around for part of the year, sporophyte that makes the spores. If we look at other plants that will, like the ferns and the gymnosperms in this case, and other plants after the non-vascular plants, we see a shift towards larger sporophytes. So the gametophyte in ferns is actually microscopic. So the gametophyte is really small and it's short-lived. So it's the total opposite. The gametophyte is short-lived and small. The sporophyte is big, photosynthetic, and long-lived. Okay, and same thing just to the nth degree with the gymnosperms. Now the gametophyte is this little teeny tiny thing that forms inside of the seed or inside of the reproductive structures like the pollen. Whereas the sporophyte is the huge big tree, right? So a tree is a sporophyte. The whole thing's a sporophyte. It's photosynthetic, it's long-lived, it's very large. The gametophyte of a tree is small, it's inside of the seed, it's microscopic and only present for a short amount of time. And again, that's a result of evolution and the pressure to make more offspring. So non-vascular plants, I just want to sort of touch on the differences of non-vascular plants compared to plants, the other later plant phyla. So we'll be talking about the traits that those later plant phyla have, but I just want to be explicit here. So first of all, gametophytes are the dominant generation. So like I was just saying, in all other plants like the ferns and the trees, the sporophyte is dominant. That's what's big and photosynthetic. But in non-vascular plants, the gametophyte is the dominant generation. That means it's photosynthetic, it's longer lived, and it's bigger than the sporophyte. In non-vascular plants, it will be the opposite. The sporophyte is dominant in all other plants. So again, sporophytes are dependent on the gametophyte. So the sporophyte is small and short-lived and has to feed off of the gametophyte. Okay, so we could say that the sporophytes are not photosynthetic. Again, they have to feed off of the gametophyte. This will be the opposite in other plants. Okay, the sporophytes will be photosynthetic in other plants. And then the third trait, and this is the one that's not related to the life cycle, is where their name comes from, non-vascular. So the non-vascular plants lack tissues for structural support and conduction found in other plants. So what we're talking about here is stems and roots and leaves are not present. And so you might be thinking, well, but those things looked leafy. While the mosses might appear to have leafy kind of structures to our eyes, if you look at them very closely, they actually don't have the vascular tissue that stems and leaves normally have. So they don't have these veins that run through them. They're not capable of moving water. So conduction is about moving water. Okay. Structural support, this is part of why they have to be so small. So the consequence of this is that they're small. They don't have any structural support to get big. Okay, so they're usually just like an inch or two high. They aren't anchored into the ground. So you can actually pick up a chunk of moss and there's nothing underneath the moss holding it into the soil. And you can just take that moss and stick it back onto another piece of soil. There's no roots that are buried because they don't have them. So they're small, they're not anchored, and they're dependent on water landing on them. Because they don't have roots. They can't pull roots up out of the soil, and so the water has to actually splash onto them. So this is why you tend to find mosses in places like forest floors where it's shady and moist, or along creek beds where they're getting water splashed onto them. You're not going to very often find mosses, for example, just hanging out in Aurora because Aurora's too dry. There's no way for them to get water without water actually coming onto them from the air. So they're really limited in where they can live for that reason. Okay, so they don't have stems, roots, or leaves for structural support and conduction. Those structures are found in all other plants. So the consequence is that they're small, they're not anchored into the soil, they can't pull water out of the soil, they have to get water from the environment. So this is actually the second dependency of mosses for water in the environment. So remember, they have to have water in the environment for their sperm to swim, for their reproduction, and then they also have to have water in the environment just to give them water because they don't have roots. So I've just kind of given you all the reasons why non-vascular plants are somewhat limited in where they can live and their ecology. But despite their limitations, they were probably like the earliest land plants and had a huge impact on Earth. So they're the ones that first started to build soil, right? And they're the ones that first started to change the environment of Earth. So one of the things that's interesting about them is that their body tissues are relatively decay resistant. So a lot of types of mosses, the moss doesn't break down that quickly. And then especially in some of the environments they live in, they end up acidic. So you may have heard, for example, of peat bogs. And peat bogs are very acidic. They're very preserving. And so that's what I have a picture of here. This is what's called a bog body. This is one of the most famous ones called Tolland Man, if you're interested in reading more about him. So bog bodies were bodies that were ended up in the bog sometime during the Iron Age, so thousands and thousands of years ago. And the bodies are preserved like so well, so you can actually see the skin of still here. The hair is oftentimes preserved because the environment is so acidic. And so the tissues of liverworts and mosses oftentimes are maintained for a really long time. And so when they are holding their tissues, they're not decaying, the CO2 that went into their tissues doesn't get re-released. So for plants that break down quickly, they absorb CO2 into their bodies through photosynthesis. Okay, so let me explain this a little better. Oops. So through photosynthesis, they absorb CO2 into their body. And then when they die and decay, the CO2 is re-released out of their body back into the atmosphere. So as long as that process is happening relatively at the same time, plants aren't really changing the CO2 in the environment. But what happens is if decay is delayed, then the CO2 is just permanently held out of the air. And this is sometimes called a reservoir, right? So they're actually holding the CO2 out of the air. And so that's what we think happened with the seedless plants in Earth's early history, is many of them did not decay. They pulled CO2 out of the environment and held that CO2 out of the air, and that had a moderating effect on Earth's climate, so actually cooled it. And so because this was happening all over the Earth for a long period of time, the effects may have been really, really large and may have actually paved the way for many later plants and animals as well.